Well, it just starts on its own now. I'll pause it real quick, okay?
So we're live, Fabian?
Yes, the
Okay.
floor is yours.
Okay, thank you. So I want to start off by welcoming everyone to this webinar. It took a little while to this GBA webinar. So this is one of a series that's kind of traveling around the globe. So this is for the North American webinar, although we have guests from Germany joining us today. I would want to, although it's probably you've probably been warned through Zoom, but I just want to remind you again that this is being recorded. So if you choose to ask questions later on, your voice will be recorded and posted on the GBA, the Global BioFoundry Alliance website. So if you go there, you can see the posts for some of the past webinars that are there. If you want to listen to them and send the link to your friends later who might be interested in watching, because they're probably sleeping now. So today's theme is around mammalian cells, and with two specific goals. One of them was to discuss or get a presentation on the technology around automation for cell culturing, mammalian cell culturing, more specifically Ipscs in this case, and how to passage, grow, expand, and hopefully edit these cells. For anyone that's tried to do genome engineering on stem cells, they realize that developing an automation process is really important in terms of getting the throughput and the numbers high up there. So we'll get a great presentation by Bastian and Yannick from Germany. And the second presentation of the webinar will be from Leslie Mitchell from Neochromosome. I'll do the full presentation later, but Leslie is going to talk to us about how to build and assemble large pieces of DNA and introduce them into mammalian cells. So at this point, I think we'll move to the first set of speakers. We decided that it was best to talk about cell culturing and automation first. So I want to introduce your two speakers. We have two speakers with one presentation. So our first speaker, Yannick Kutz, I hope I pronounced that properly, comes to us. Yes, he's grinning, so obviously I butchered it. Comes from the Life and Brain Institute, which I believe is a product of the University of Bonn in Germany. Yannick did his PhD at the Life and Brain, worked on CNS drug metabolism and human iPSC cell-derived neural spheroids. And also given part of that presentation is Bastian Nissing. Bastian now works as a group leader in the automation and life science in the Fraunhofer Institute for Production Technology. And he obtained his degree from the University of Stuttgart in medical technology as a Master's of Science in Biomechanics and Biomaterials. So on that, the floor is yours, Yannick and Bastian.
Okay, thanks for the kind introduction. So let me share my screen. So can you see it?
Yes.
Okay, perfect. So again, thanks Martin for the kind introduction, and thanks for giving us the opportunity to present our work about the stem cell factory in this webinar. So my name is Yannick Breitkreuz, and together with Bastian Nissing, we want to give you an overview about automated generation and expansion of human iPS cells. So, but first, let's briefly introduce our company and institute. So I'm working at the Life and Brain company. It's a life science company which was founded 2002, and it is serving as a commercial hub of the University Hospital of Bonn. The company is divided into 3 platforms. So the Celomic unit where I'm working, with a strong focus on human pluripotent stem cells for the controlled differentiation into neuronal and glia cells. And beside this platform, our company includes the genomics and the electrophysiology services. Our aim is to close the gap between university research and business. In academia, translational developments frequently get stuck at an early stage because because insufficient know-how and lack of financial sources prevent the development of a proof of concept required for further downstream commercialization and market entry. So the life and brain act as a revolving door between academia, research, and industry. Innovative research findings are recognized early and developed further into marketable biomedical products and services. So our mission within the Cellomics unit is the generation of iPS cell-based products and services for diagnostic and therapy of nervous system disorders. We get samples of different patients. We reprogram these somatic cells into iPS cells. And if necessary, we can modify these cells by genome editing, and subsequently we differentiate the cells into neuronal and glia cells in 2D or 3D. So on this slide, you can see an overview of our cell portfolio. Started from iPS cells, we can produce on the one hand cortical neurons, for example, dopaminergic neurons, as well as astroglia or microglia cells via conventional 2D differentiation. And we also established protocols for forward programming differentiation of, for example, glutamatergic and GABAergic neurons. On the other hand, we are culturing 3D organoids and spheroids with the main focus on the forebrain. Now I would like to turn over to Bastian, and he will introduce the Fraunhofer EPT.
Yes, thank you very much, Yannick. So the Fraunhofer Institute for Production Technology is an institute located in Aachen in Germany. We have nearly 500 employees. We have a huge machine hall and a lot of laboratories. We was founded in 1980, and we have an overall budget of 30 million euros per year. In our institute, we have 4 main departments. And on the next slide, you can see the main department where I work, so the Department for Production Metrology. And within this department, we have 3 different groups. The group for optical metrology and imaging methods, the group of for digital infrastructures, and the group of automation and life science, where I'm the group manager. And within this group, automation and life science, we had many experience over the last years in lab automation. And I think on the next slide, we can see how our approach is if we want to go for an automated process for cell cultures. So our approach is that we go for a platform technology, and this platform is a fully automated closed system. We have a highly modular and flexible system, so we can define different use cases and make an automated platform for this. And we need to be fully integrated solutions, or it's because otherwise, if we don't have a completely fully closed system, it is not GMP compliant. And because we produce the cells under GMP conditions, we need a completely closed system. And here on the next slide, you can see how much experience we have over the last nearly 20 years in cell culture automation. So it started all in the year 2003 with the so-called cell host. This was a cooperation with the company of Hamilton, where we built a liquid handling unit. And here you can see on the left and on the right side 2 incubators. And then we only we automated the process for media exchange and then automated incubation. Then there was a development of the so-called biolevitator also with Hamilton. And then nearly 10 years ago, we started with the stem cell factory, where we wanted to produce in an automated way induced pluripotent stem cells. This is also what we're going to tell you later on. And then at the moment, we have at our institute and at the Institute of Yannick 3 different platforms, the so-called stem cell factory, the stem cell discovery, and the autostem platform. And with those platforms, we can produce in an automated way induced pluripotent stem cells and mesenchymal stem cells. And now I will hand over to Yannick again.
Thanks, Bastian. So now let's go into the most interesting part of our talk, so the presentation of our stem cell factory. So the use of human-induced pluripotent stem cells offers fascinating perspectives for disease modeling, drug screening, and personalized therapy. The globally increasing demand for patient-derived iPS cells has created an urgent need for standardized and automated production processes. To that end, we developed the Stem Cell Factory, a modular platform for automated generation and expansion of human iPS cells. So in this example, you can see that the robotic arm places fresh media and the plate of interest onto the liquid handling unit, and a classical media change of a 24-well plate is performed. Next, with our high-speed microscope, we can image whole plates in less than 2 minutes, followed by automatic quality validation. By using the cell selector, we automated clonal selection and expansion of single cells or colonies, which we can perfectly use for our new developed processes. So as you can see here now in this figure, you can see the stem cell factory includes different modules. In the movie, you could already see the liquid handling unit, the automated clone picker, and the high-speed microscope, which are all connected to automated incubators, material hotels, and analytic tools by a robotic arm. Furthermore, we implemented a centrifuge and a Pacol station, as well as a nucleofector in the last couple of months or years. But we will present all of these single modules step by step during the next minutes. In general, as you can see on the right side, the entire unit is encased in a clean room cabinet. So next, Bastian will show you how it all began.
Yes, as I already mentioned, we started with this project 10 years ago. And as we start with the reprogramming of fibroblasts. So here you can see the whole process we fully automate in the stem cell factory. So we start with fibroblast, and then we do the reprogramming step, and afterwards we do the clonal expansion in 24-well plates. And the special thing is that we do a confluence-based splitting or media change. So we have a decision. If we go for the confluence over 80%, we do a split, or if we have it under 80%, we do a media exchange. And then we can produce these cells and can scale it afterwards up in a 6-well plate format. So this is the whole process we started 10 years ago. And now I will go through the different devices we need for this process. So first of all, the high-speed microscope. This was an invention of our institute. So the special thing on this microscope is that we are able to scan in the movement. As you can see here on the video, we can put the whole plate in the microscope, and then now you can see how fast we can scan. So the advantage is that we scan in the movement, so there's no swapping of a liquid, and without this swapping of liquid, we get focused images. And because of those focused images, we can analyze those images. and can use the results for the process optimization and for the process control. So we can record about 300 square centimeters per minute with a 5 times objective. As I told you, we need this high-speed microscope for the analysis of the images and the results for the control of the production process. We invented also a completely algorithm, a deep learning algorithm for the detection and classification of induced pluripotent stem cells. So we made 6 different classes, as you can see on the right side. So we have in black the background, we have in blue single IPS cells, we have in gray the IPS colonies, we have in purple dead cells, in red differentiated cells, and in green the unknown class. So what we can do is when we make this image of the whole well, we can also analyze the whole well and make this classification of the different cell classes. And then again, can we make a decision for the process control, what we're going to do on the next step. And also what we can adjust is the daily cell culture monitoring, and not only on few points, but within 100% process control.
Good. Then I will go on a little bit more into the automated reprogramming process. So on this slide, you can see a high-level workflow chart for automated reprogramming of human fibroblasts into iPS cells using the stem cell factory. And as Bastian already mentioned, the process is split in 3 phases. So the reprogramming phase, the clonal expansion phase, and the expansion phase of the chosen clones at the end. So while the reprogramming phase is in an inflexible sequential procedure of protocols, the expansion phase is a dynamic cell growth orientated phase. And during the whole process, we are working in 6 and 24 well plates. So what you can see here now in A, the key time points and processing steps of the reprogramming protocol, and the reprogramming efficiency ranging between 0.6 and 0.8%, and this is comparable to the reprogramming efficiencies reported by the manufacturer. So this is about 1%. And in C, you can see representative whole well images of single wells of a 6 well plate at day 21 post-infection, and this show cell colonies in black and cell-free areas in white. So a key step during the protocol is the clonal expansion. For this, we integrated a cell selector. The software can automatically select the colonies that meet specified conditions. And for each colony, the robot arm takes a fresh tip, then goes to the colony of interest and scratch the defined area, and then place the cell into a 24 well plate. These steps are performed within about 30 seconds. So the success rate is comparable between manual and automated picking, with an advantage for automation in attachment. So next you can see the long-term expansion of the human iPS cells. The human iPS cell lines expand via the stem cell factory, show constant and exponential cell growth over 10 passages. In addition, flow cytometry analysis was performed after each passage and showed a high expression of a typical marker TRA160 in all evaluated cell lines. Furthermore, representative phase contrast images of these reprogrammed iPSC lines at passage 10 are shown here in C for 5 different cell lines. So in general, we can reprogram, cultivate, and expand up to 40 6-well plates with theoretically up to 240 cell lines in parallel per day by using the stem cell factory. And just for your interest, let's say a good technician can handle 8 to 10 reprogramming protocols in parallel. So next, Bastian will show the newest modules on the stem cell factory.
Yes. So as I mentioned, we started 10 years ago, and as it wasn't enough for us to only reprogram fibroblasts, we started to implement 2 new processes on the stem cell factory in the last years. So the first new process is the reprogramming of erythroblasts. So we can use patient blood samples for the whole reprogramming process. This is much more easier for us to handle and also to get these probes, because a tissue sample is it's not that easy to get in tissue sample. And the second huge new step on the stem cell factory is the automated genome editing. So we also added the automated genome editing on the stem cell factory. And now I start with the reprogramming of the erythroblasts. So what we did so far and what we developed is so-called patrol station. On the left side, you can see it. This patrol station can be handled automatically by the robot. So we put a tube inside of the patrol station. Then we have a camera system and an illumination system. And with this system, we make an image of the patrol phase. And then we can automatically detect this patrol phase. And once we have detected this patrol phase, we can easily calculate the volume and the height of this patrol phase. And if here on the right side, you can see an example for the volume of a patrol phase. And with this, we can easily give those 2 parameters to the liquid handling unit. And then we put the tube with the robot also to the liquid handling unit. And then we can define on which height, which volume the liquid handling unit has to pipette. So we get the patrol phase. And here on the downside, you can see the experiments we did manual and automated. So as you can see, in some cases, we even get more cells than in the manual process. In some cases not. So I think we are quite equal to the manual process. But what you could see as we did the FACS analysis of the cells, that in the automated way, the cells were more, the markers were more positive than in the manual way. So the automated way was quite better than the manual way. So, and as we have implemented this on the platform, we now can use blood samples for the reprogramming of cells. And then we added also the automated genome editing on the next slide. What we did is that we implement a nuclear factor from Lonza on the deck of the liquid handling unit. And theoretically, we are able to make 96 wells or 96 genome editing by one. But the thing is that we can't handle so much plates on the liquid handling unit. So we are limited by hardware to 24 wells. So we can handle 24 RNPs with 24 cell lines in parallel by one shot. So we need about 20 to 30 minutes for this whole process. And in the middle, you can see the data of GFP construct we added it to the iPS cell lines. So we also go for the clonal expansion of the cells in 6-well plate. So this is because we need so much space on the liquid handling unit. So we need for 24 wells, 4 6-well plates, and then we use one 24-well plate for the quality control, where we put the cell bulk, the rest of the cell bulk in it. And as you can see in the middle, we successfully edited the GFP construct in an automated way and also in a manual way. You see it on the right side, and also the efficiency of the manual and the automated way. So the automated efficiency was at least a little bit higher than the manual way. So as we could show here, we could, or we had successfully automated the whole process of genome editing on the stem cell factory. Now I want to give you a short summary of our presentation. So what have we done so far within the project stem cell factory? We had successfully automated reprogramming for different cell sources. So we can use human fibroblasts and we can use blood samples. We have a highly efficient clone picking device where we can pick about, in about 30 seconds one clone. We have the parallelized clonal expansion of human-induced pluripotent stem cells. And we have this deep learning image analysis developed where we can classify the images of the whole well or of the whole plate. And additionally, we have added the automated genome editing on the platform. Now I want to thank all of the partners and all of the people who was working within this project, especially the Life and Brain and the Institute of Reconstructive Neurobiology, and also the Helmholtz Institute in Aachen, and also the people from my department. And if you have any questions, feel free to ask.
Great, thank you, Bastian and Yannick. At this point, the floor is open for questions. Fabian, I think people can ask their questions, or does that have to be into the chat?
You can do either. You can send the questions to chat or Q&A, and then you can also use the raise the hand feature, which will bring you to the top, and then I can unmute whoever wants to verbally ask a question.
Okay, so the floor is open for questions if there are any. Well, I have some questions. I have one question that's really intriguing to me is, so you mentioned that you can get up to 40 6-well plates, so 240 cell lines in parallel. How do you, and I guess there's a scheduling aspect of this, all the cells are not synchronized, so do you have to image and make a decision on each cell line on a daily basis? And if that's true, do you have to make decisions on a daily basis as to who gets passed or split? And how does a schedule work around that?
So we use a prioritized scheduler. So we have different options and different operations that have a different prioritization. So for example, we scan the whole plate, and then normally we do a confluence per well, and then we check if more than 4 wells are above this this threshold, and then we do a whole plate media split or media change. Okay. So otherwise, if we do it with single wells, yeah, we couldn't handle it.
Okay, so you make some inferences based on a few. And do you find that from your reprogramming, you most likely have all sorts of different growth rates? Do you eventually kind of home in and standardize on a certain set of cell lines that you can synchronize more or less, or?
So at the moment, the cell lines we use, I think they are more or less synchronized to each other. But yeah, at the moment, we don't have that much issues with the scheduling problem, because when we have this threshold about of 80%, and there are 4 wells above this 80%, we just take all the whole plate. Yeah.
Good.
Yeah, and so it could be possible that we also could use more than 40 plates, but we calculated with the worst a case that, for example, all 40 plates has to be splitted on one day, and more or less the splitting process is a little bit time consumed. So if you take the plate, do the microscoping thing and then splitting, it's for one plate, it's around about 30 minutes. So 27 minutes. So this is the reason that we said, okay, if we have a 20-hour day, we can do this for maximum 40 plates. Yeah.
I see. There's a question here, two questions from Smita. So do you passage the IPS as single cells or as clumps?
As clumps? Sorry?
Do you passage the IPS as single cells or as clumps? As far as I can tell, it's clumps, right?
Yeah, so we have both options, but normally, of course, so we take it from the the cell selector is scratching the colony of interest, and there's no accutase step or something like this inside. So it's more clumps, yeah.
Right. And Smita also had a question, same question as me. So on your editing, I realized you guys just, I guess you onboarded that not that long ago. But how do you select your clones from your editing, and how do you genotype afterwards?
Yeah. So this is yeah. So we are doing this at the manual step. So the QC is doing manual at the moment. So at first we have the 24-well plate where we have more cells comparable to the 6-well plate, and we take the 24-well plate out of the factory. Then we are doing the sequencing, and then we can see the indel rates. So if for example we have a high indel rate, we can be sure that more or less, let's say that 70% of our colonies or 80%, it depends a little bit on the RNP, is positive for our gene of interest. So, and then we decide how many colonies or how many colonies we will select from the 6-well plate. And for example, let's say in the worst case, and it's a RNP where you just have a editing efficiency of 20%, then we would choose, let's say about 10 to 20 or 10 to 15 colonies, and then we analyze all single colonies so that we can be sure that at the end we have monoclonal edited cells. But these steps of the QC more or less is manual outside the factory.
Good. I suspect Leslie will teach us how to genotype lots of cells quickly. Well, on that note, just to make sure that we don't go too much overboard, I want to thank you guys, Bastian and Yannick, for a great talk. I actually learned a lot, and hopefully we can keep the discussion going as we try to set that up over here and try not to replicate all the mistakes you guys made.
Hopefully not.
Thank you very much.
So thanks again.
Yeah, you're welcome.
All right. So we're going to move on to our second presentation for this webinar, which is going to be given by Leslie Mitchell. Leslie is a Canadian, so it's fun to have a Canadian as part of this webinar. Leslie obtained her PhD from the University of Ottawa, and then went on to do a postdoc first at where were you first? You ended up in NYU, but you were first at
Hopkins.
At Hopkins, right? And then since then has moved on from the postdoc to co-found and is now the president of New Chromosomes since 2019. And Leslie was part of the synthetic biology, the SC 2.0 for Saccharomyces cerevisiae, building genomes there, and has moved on to mammalian cells and mouse cells, I guess is what we're going to hear about today. So the floor is all yours, Leslie.
Thanks, Vince, and thanks for everyone for being here. And thanks to the previous presenters. That was kind of a tour de force on building up a system on something that's really hard to do. I would have loved to hear a little bit about the LIMS system you guys coupled to that, but maybe that's a conversation for offline. So I realized when I was putting this together that I'm coming up on my 10-year anniversary on thinking about genome writing. So that's exciting. I'll talk a little bit about how I got into this. Vince kind of referenced this, and that was in my PhD, I was working on yeast genetics, and I invited the great Jeff Buca to be an external advisor on my thesis committee, where I learned about the yeast genome project that far predates me. And Jeff sort of asked the question one day at a casual lunch with some colleagues and said, what would it take to write a designer yeast genome from scratch? And that's a kind of a big problem, a 12 million base pair problem across 16 chromosomes, 6,000 genes, and of course, centromeres and telomeres. The idea being, can you bring that sequence into a computer, redesign it, and spit out the other side a synthetic cell carrying a designer genome? 15 years later, of which I have been into this for 10, many, many institutions, many, many scientists, and who knows how much money. Unlimited learnings have come out of it, I would say. It feels like a credit card commercial, but the idea that technology development underpinning this project is really what I'm going to talk about today, and how we've sort of taken the learnings from SC2, some of the summary kind of conclusions of this project, which is nearing completion, are shown here. The idea that with a centralized design, distributed collaborations are successful, including across almost all of the continents on the planet. Those type of collaborations can successfully be used to undertake these big projects related to genome writing. Even at the 12 megabase scale, that's a pretty big project. At this point, the project has functionally demonstrated the entire design works in cells, and it's a question of consolidating chromosomes into a single cell. And then out the other end has come this formalized technology stack related to what I've started to call Genome-Scale synthetic biology, which I will just make the point, it's distinct from plain Jane vanilla synthetic biology, if you will, which is to say it's typically gene at a time or a few genes at a time with a sort of a different requirement for technology related in this design, build, deliver, test, but customized in the context of thinking about how do you optimize sequences, not just at the base or gene level, but rather make sure that every base change one makes is going to be compatible globally when you're changing sequences across the whole genome. How do you then, this is the big piece, the build and deliver, how do you start thinking about sourcing all of that synthetic DNA, making pieces that are meaningfully large to put into cells, what types of cells you're going to go into, what are the challenges related to those different types of cells. Yeast and mammalian cells are like worlds apart in terms of putting large pieces of DNA into them. And then what kind of analytics stack do you need at the end of the day to accommodate measuring and ultimately debugging, which is not shown on here. I guess the bottom line to this, to the point of this slide, is that one would need a combination of software and wet lab processes at a fairly substantial scale, and a good amount of automation to the interest of this group today. So in the context of SC2, we've sort of put together this framework for thinking about this standard synthetic biology workflow at the genome scale. This matters really because I think two things. One, most genomes are far larger than the commonly available tools that are in kind of standard use across academia and industry. And what only really occurred to me over the past couple of months is that we're sort of stuck in this sort of self-fulfilling prophecy in the context of thinking about how to engineer cells. And that is what's shown down here, which is a little bit hidden in this slide, but the idea that de novo gene synthesis is typically limited to the order of 5 kb. Designer nucleases are really most effective at the base pair or a couple base pair change level. And we could have arguments around that. And then viral gene delivery is really focused on the 5 to 10 kb range. And so with this sort of self-fulfilling loop of making DNAs that are compatible with the size of viral vectors we have for delivery, we're only really touching a fraction of all of these like commonly manipulated genomes. And so starting to think about this at these broader scales, I think is really important. And what kind of technology development do we need? This has been formalized in the context, I'm sure a lot of you are very familiar with the GP Write project. I think Amy Schwartz is even on the call today. And this idea that there's a whole host of technology challenges related to writing genomes. This was all formalized by Neely Ostrov and a whole group of really wonderful people a couple of years ago in this white paper. So today I'll just talk about two key aspects of SC2 that we've taken sort of learnings from and converted to thinking about how to apply them in mammalian genomes. And that is scalable DNA assembly methods supported by automation. And then what I'm calling delivery, which is the endless challenge of how do you get efficiently DNA into cells, in particular mammalian cells. So to tackle the assembly problem, we started with how we sort of thought about SC2 DNA delivery, and that was this process we called swap-in. So we built up to 10 kb segments of DNA, starting with either oligos or 3 kb parts. These were designed in a way that they had termini that were unique restriction sites, enabling directional assembly. So in vitro, ligating together a series of 10 kb parts, and then transforming into yeast using a series of selection tricks to replace segment by segment portions of the yeast genome. So this idea that yeast is incredibly good at homologous recombination can be leveraged. What we didn't like about this approach is that it really meant that the DNA was, first of all, yeast specific, but second of all, stuck inside of a yeast chromosome. And so we converted this technology for extra chromosomal DNA assembly and thinking about driving efficient assembly of 20 or 35 kb fragments simultaneously, which really comes down to what sequence do you encode in the termini, and then leveraging this iterative kind of selection strategy such that we can grow these sequences from 30 to 60 to 90 via these iterative selection steps and transformation and sub-assemblies. So this swap-in approach I'll highlight in the context of a publication that recently came out, but that's one way that we're starting to think about assembling longer DNAs, and that's leveraging yeast and the learnings of SC2. So the second big challenge is how do you get these large DNAs into mammalian cells? We spent a couple of years working on artificial chromosomes, and the challenge there is really this big hairy issue about centromeres in mammalian cells, which are long and repetitive and complicated, and only recently has the primary sequence of these things kind of been unveiled. And so our approach has really been to think about integrating these long DNAs directly into resident chromosomes. And so the first sort of tack we took on thinking about delivering these long DNAs was using a technology that pre-exists and had been established for studying differentiation of stem cells via, the very quick integration of inducibly expressed transcription factors called inducible cassette exchange, or ICE. And so we working together with Michael Kaiba, kind of grabbed this technology, which had been developed for mouse ES cells, and started asking questions about, in the context of this delivery, which relies on inducible Cree expression, recombination into a pre-existing landing pad of this sort of complicated designer DNA with a payload, how long of a payload length can we actually achieve for efficient delivery, where the resulting cell line is readily selected with an inducible GFP signal and resistance to neomycin. So those are the two sort of technology pieces that we started to think about, and we placed them in the context of assembling a human gene with the goal of delivering it to mouse cells. I would note that in terms of genome writing, this is probably one of the first areas that has already been commercialized. Obviously, humanized mice are leveraging these sort of long pieces of DNA. It is an extremely robust technology at this point. And so we picked HPRT. If you're familiar with that gene, it's a selectable gene in the context, selectable and counter-selectable, but it's an enzyme that has a key role in purine salvage. We looked at the gene and kind of arbitrarily decided that it was 102 kb in length, looking at some of the regions of CTCF binding sites and other regulatory kind of elements that might be important. We chose to call the gene boundaries 60 kb of the gene and then 40 kb up and downstream. So in terms of thinking about using that e-swap-in technology for assembly, we segmented out this gene into approximately 3 kb fragments. And those then could be sourced via a number of mechanisms, but as we all know, synthetic DNA is a little pricey and sometimes doesn't deliver on the promise of having that sequence arrive in your lab. And so we sourced these 3 kb segments via PCR from a cell line in the lab. These fragments, we then use the e-swap-in technology that I described in 3 steps, assemble this 102 kb sequence in about 6 weeks using yeast. It was assembled in the context of a shuttle vector, meaning it could be recovered into E. coli. In E. coli, one can take the product of each of these 3 steps and digest it and show the correct patterns. It can also be sequence verified and also amplified for the ultimate next step, which is delivery. I'll also point out at this point, one of the genotyping strategies we use extensively in pretty much all of our workflows for genome writing is genotyping Pcr. So I'll just highlight that really quickly. We call it Junction Pcr. So in the context of these assembly steps, the idea is that in yeast, in a colony that forms following an assembly transformation, if you design primers that span the junction between 2 parts that are supposed to be assembled, the only way you'll get a PCR product out of that is if the assembly has correctly happened. If you check the assembly junction at every single point along, upwards of 30 fragments, you can very quickly identify putative winners, those that carry putatively the correct assembly. Notable for this community, this is a picture from the NYU lab and Jeff's lab. We put together an automated platform for screening yeast colonies that essentially is a high throughput qPCR platform operating at 1,536 PCR reactions at a time at a very, very low cost. I will note that that technology was spun out for use with COVID screening. Excuse me, and at this point, almost the majority of New York COVID tests are actually run using this pipeline at this point. But separate to that, originally developed for genome writing, which is exciting. This is the platform that we use for evaluating both yeast colonies as well as the downstream delivery step, which looks like this. So we take the product of the yeast assemblies, recover to E. coli, prep a lot of DNA, and then transfect that naked DNA into the mouse ES cells that are sort of pre-populated with the landing pad. The DNA that we built is correctly designed such that heterotypic lock sites flanking this Cre recombinase that's inducibly expressed will lead to recombination, which will overwrite the Cre and introduce a GFP downstream of the inducible promoter and reconstitute the neomycin, which is otherwise lacking a start codon, to the extent that one can look for colonies that become green and have resistance to neomycin or G-phonate on the plate. One can do this high throughput genotyping. In this case, we are introducing human DNA into a mouse cell. So we could just evaluate the presence of this human DNA. I'm not showing you the control reaction, which is a bummer because we always say no control out of control. But in this case, you'll have to just trust me. You can also evaluate the new junctions that form in the genome, which we're showing here. We took clones from this experiment and looked for the presence of the human protein being expressed. An unmodified cell has no band in this Western blot, where one clone, although we've done this with multiple clones at this point, indeed shows a band, suggesting functional reconstitution of a full-length human gene in a mouse ES cell via this method. To evaluate the genotype or the karyotype of these cells, we also did some metaphase spreads and showed that the karyotypically the genome was normal post-delivery. And then for the stem cell experts in the room, so to speak, the virtual room, we also evaluated a series of markers important for stemness, I will say, and showed that there was no sort of negative effect on the stem cell potential of these cells as a function of this genome modification. So the next question was, I'll just go back to this slide, the product of this delivery, while very exciting that it's this sort of relatively high efficient method for getting these up to 100 kb DNAs into cells, and we haven't really gone high, well, I can't speak to the efforts. Indeed, we've shown larger deliveries than that at this point, but you're left with these big scars on either side of the delivered DNA. So another effort that's been ongoing is to design a Scarless delivery system, nearly Scarless, I should say, where the product of the heterotypic recombination leaves just the lock sites and eliminates all of the selectable markers. I'll just point out this is achieved through the use of selectable counter-selectable markers, thymidine kinase or PIG-A, which is a very interesting gene that can be selected for and against in cells incredibly efficiently. And so the idea is with a single step, one can deliver the landing pad into a locus, select for the introduction of pyrimycin, inducibly express TK or PIG-A, and then similar to the previous work, come in with a payload DNA, and you can do this one of a couple ways. If your payload has selectable markers outside of the heterotypic lock sites, you can have a transient selection for the blastocide and GFP, which allows you to select for cells that have actually had DNA transfected, but ultimately one counter selects TK or PIG-A to achieve single step Scarless delivery. So I hope that's clear, but if it's not, both of these, all of this work has recently been published. I know the HPRT story sort of describes some of the automation and some of the platform development. And then I would highly recommend the BigIn story, which is the one in PNAS, for people interested in thinking about delivering anything CRISPR-based or anything DNA-based to cells. There are some really solid workflows on how to think about off-target effects that you might not otherwise be thinking about. Where do all these vectors go? They don't always get lost, and so it's really important to screen for them up front and make sure you're picking clones for these next steps that are actually absent of any vector and any other junk that you don't really want in the cell. So all these workflows are described. A lot of this in Jeff's lab has been formalized in the context of this project called the Dark Matter Project. The purpose of this is to start to understand the function of non-coding variation in the human genome. It's also, thinking about how regulatory elements impact gene expression. And so also encourage you to check out this project. At this point, there's like 20 different loci across a lot of different organisms that are in play to start thinking about these long DNA delivery projects. A lot of it is going into mouse and a lot of it is cloning mice to understand some pretty sophisticated phenotypes. I will put in a small plug for NEO at this point. So this idea that genomes are the basis for all biotechnology products listed over here, viral genomes underpin gene therapy and so many more things. Microbial genomes run the gamut from therapeutics to food to agriculture, and mammalian genomes underpin cell therapies, regenerative medicines, et cetera, et cetera. the thing that nobody really steps back to think about is that naturally occurring genomes are evolved and not designed. So evolution has driven all these weird characteristics that are sort of the product of chance. There's no reason, now that we're starting to develop these genome writing technologies, to believe we should be limited to what nature gave us. So this idea of writing totally designer genomes from scratch is really the purpose of NEO. We're thinking about projects across the gamut, viral genomes, microbial genomes, mammalian genomes. I will put the caveat that in the mammalian sphere, we're definitely thinking about particularly critical parts of the genome rather than the whole thing on day one. But I think that's the direction that we should all be thinking about. So I'll stop there. I'm happy to take any questions. The NYU team is fantastic, and I miss them a lot. Luckily, Jeff and several of these other colleagues are starting to, Jeff is a co-founder of NEO, but several other colleagues have jumped ship and joined NEO. It's a growing team. We're hiring. If anyone's interested, please reach out. So thank you very much.
Thank you, Leslie, for a great talk. We're all going to read those papers for sure. Before I ask all my questions, I'm going to open it up to the floor for questions if there are any. All right, no questions. I'm going to ask my questions.
Okay.
It's going to be a Leslie Vince discussion. What's the efficiency? So your transfection, how do you do that? What kind of efficiencies are you getting?
Yeah, it's just electroporation right now. The efficiency is, I think it's like 5 million cells or something like that. And we're getting like 10 or 12 clones pretty reliably. It is not, you know, gangbusters. This is not ready for prime time on delivering libraries, for example. To that point, this idea of these sort of amazingly sophisticated automation platforms that would enable some of these deliveries in series, essentially, in parallel, across these platforms would be highly valuable. I think they're also super compatible with these selection techniques that are incredibly clean. And so this sort of medium-sized miniaturization and high throughput automation could take all of this really to the next level.
Yeah, I think you're right. But getting these efficiencies up is good, or else you're just handling too many clones or searching for the needles in the
One thing I'll say is that the HPRT publication, we've done a lot of optimization on the delivery step at this point when we're using ICE. And so we're at the point where almost every clone we get out is what we want. So we're not getting a lot of clones, but it's nearly 100%. And I think that's an important point to make, that even if you have 10, you're in a fantastic position because it's like 90% or 100% correct.
Right. I'm just thinking about, because you're using markers quite a bit, and to eventually move to markerless systems, that number's gonna have to go up. What about, so, and because you're using these markers in these systems, what about because I guess it limits you to the site where you're going. So what about things like, say, fiber? Like if you take these stem cells and you differentiate them, what actually happens to these loci? And
I mean, I think it depends. So in theory, another reason I didn't really highlight, in addition to the scars associated with ICE, it's not really easy to move the system around the genome. And we can talk more about that, but the big in technology is designed such that you can put the landing pad anywhere. And I realized that one of the big sort of challenges associated with differentiation is once the genome starts to undergo all these gymnastics, if you will, a lot of loci just get silenced. And so it's important to be able to take, I don't know how many shots on goal you need to find the place where the expression happens, but that idea that you have a portable landing pad and it's readily introduced into, I would imagine, most places except centromeres and other repetitive regions in the genome, you'll be able to design, hopefully, an experiment that will get the results you want. A lot of what we're doing, I should say a lot of what Jeff's lab is doing, is functional replacement. So delete a locus with the landing pad and then come in to replace, sort of deliver the designer version of what used to be there and test function relative to wild type.
Okay. Yeah, I keep thinking like the platform that we heard from Yannick and Bastian before,
Yeah.
and what you're doing. So you could easily use the platform to search for safe harbor spaces
Totally.
for iPSCs, and kind of have a library of safe harbor spaces depending on what you want to do with your stem cells after. There's two questions from Smita here on the chat. Can you do CRISPR-based joining of chromosomes or payload of foreign DNA similar to a single chromosome in yeast?
That's a great question. I'm just going to guess yes, but I don't really know the answer.
Okay. That's a good
I'm going to
guess.
guess it would be harder than yeast because that's just a general answer also.
And our workflow is generated from embryonic stem cells directly adaptable to iPSCs.
Yeah, so that what I haven't talked about today is we've been using a lot of these approaches in human iPSCs as well. And so different challenges, mouse CSCs in particular are like very accepting of these large DNAs. The human iPSCs are like, they're quite a bit more finicky and like die on contact when the DNA hits them. So we've done some work to optimize that. That's been done in the company. And just sort of, I think it's sort of public domain information that we're thinking about sort of custom versions of the HLA locus. And so that's an area of development with successful delivery, I think of up to like 30 or 50 kb at this point and pushing up to 100 in the near term, using this big in kind of landing pad technology approach. Delete the resident locus and then bring in a custom one as a second step.
Cool. Okay. Any more questions from the floor? If there's no more questions, we've been at this for almost exactly an hour, so we're on schedule. I want to take the time again to thank Yannick and Bastian and Leslie for two great talks, and again to remind everyone that the talks are going to be posted on the GBA website very, very soon, and I think you're more than welcome to. I certainly will contact the speakers later on because now I have all these ideas bouncing around in my head. So, so thanks again for the great talks.
Thanks, Vince, for organizing.
All right.
Yeah, thanks to you.
Thank you,
Thanks a
Yannick.
lot.
Bye-bye.
Bye.
Bye.